Last but not least, the special senses. So when we say special senses, we're talking about taste, we're talking about hearing, we're talking about vision, equilibrium. Yeah. First things first, let's talk about the eye. And again, the idea with this type of lecture is that we focus on the actual physical parts and touching and feeling them, which we don't have access to, and it's a true shame. But I'm going to do my best to show this to you in as good a way as possible. All right. The lacrimal apparatus. The lacrimal apparatus. What we have here is an eye, and on the lateral aspect of that eye, there's a gland called the lacrimal gland. That lacrimal gland produces a fluid. It flows over the eye, nourishing and cleansing and cleaning the eye. And that fluid flows into these lacrimal punctums, into a lacrimal sac, and then down into the nose, basically. It's nasolacrimal duct, goes down to the nose. What I would like for you to be able to see here is this lacrimal sac where the fluid comes to, and this lacrimal gland where the fluid comes from. I will show you this. I have a nice eyeball in the lab. This will be on your test. You're going to be labeling it for me. Okay, you need to be able to see this and know what we're dealing with. Lacrimal gland, lacrimal sac, fluid produced here, washes across the eye and cleans the eye, and then this lacrimal duct or lacrimal sac on the other side. Now, you blink reflexively about every 3 to 7 seconds, and this basically spreads fluid across the eye, forcing it into these lacrimal punctums as a constant flow. And you can see a lacrimal punctum here. All right, good. Here are more vision accessory structures. So what we have are eyebrows, eyelashes or eyelids, and the conjunctiva. The way this works is as follows. The eyebrows, all right, shield the eyes from the sun and also funnel sweat away from the eyes. Think about if you're, chasing that gazelle and you got your spear, you don't want your sweat in your eyes, so you want to funnel that away from them. That's why we have eyebrows. You have eyelashes or eyelids. The lashes attached to the eyelids are very well connected to the nervous system, and they're set up on a blink reflex. So if you touch the lash, the eye instinctively closes. Yeah, it's a protective measure, if you will. And then we have the conjunctiva. So you know about the conjunctiva. You know about conjunctivitis being pink eye. The conjunctiva is a mucosal sac that connects the eyelid to the eyeball itself. What you might say the eyelid to the sclera of the eye. And then obviously you have your 6 extrinsic eye muscles. The idea, no pun intended, the idea here is that you have a superior rectus, makes the eye look up. An inferior rectus makes the eye look down, a medial rectus makes the eye look left, and a lateral rectus makes the eye look right. And then you have a superior and inferior oblique, and they twist the eye. Okay, they can twist the eye a little bit. I'm not going to ask you a whole bunch of detail off of this. I just want you to know that these exist. You do have a superior, inferior, lateral, and medial rectus muscle, and then you have these obliques that make the eye twist. And that brings us to the meat of the conversation. Now, this is important, okay? This is very important. You will be seeing this on your lab test. You will see this on your lecture test. So I'm going to go through it quickly in lab, and when we get to lecture, I'm going to go into all sorts of detail. The idea is this. On the outer portion of the eye, we've got a cornea and a sclera. The cornea is the clear part on the outside surface. This is incredibly important. If you don't learn anything else about the eye, you need to understand that the clear cornea on the outer surface, this clear part here, like where a contact lens would fit against the eye, that's the cornea. That clear part is the main light-bending apparatus of the eye. When light comes in, the lens that's in the eye simply fine-tunes the image. The main focus is done by the cornea, so you need to be friends with that, all right? And then the sclera is the white part of the eye, and to sclerize something means to harden or toughen it. The sclera is incredibly tough, incredibly strong. Below this, we have the actual physical eyeball. So let's talk about the choroid, the ciliary body, and the iris. These are considered parts of the vascular layer of the eye. The choroid is incredibly important here. The choroid is going to be what houses all the blood vessels that the eye uses for general function. So all of like your optic nerve coming in and connecting to a bunch of photoreceptors and things along those lines, none of those would function without the choroid providing blood supply. Okay, it's very important. Providing blood supply for the eye is the choroid. Sclera to choroid, and the next layer in is the retina, which we'll get to in a second. Okay, so other vascular things, the ciliary body. The ciliary body surrounds and holds the lens in place. The idea is the ciliary body is muscle, ergo it has to be vascular, it's got blood flow. The ciliary body can squeeze the lens and make it round, or stretch the lens and make it quite flat. And then is the iris. The iris is also muscle, ergo it must be vascular. The iris is what makes your pupil. So the iris is the muscle that surrounds the opening of the eye. So if you look at the colored portion here, this kind of like a bluish, if you will, maybe greenish iris there. Do I have a better picture of an iris? I don't guess I do. If you go and you look in the mirror, not the pupil, but the part around it, that's the iris. And it's capable of contracting and limiting the amount of light that can come into the eye, or dilating and increasing the amount of light that can come into the eye. All right, next, the retina. So when you look at the layers of the eye, you have the sclera, which is the hard outside, the choroid, which is that vascular layer that provides a bunch of blood supply, and then internally to that is the retina. The retina is what houses what are called the rods and cones of the eye. The rods and cones are the physical receptors themselves. They are phototransducers is what we call them. They transduce light into nervous action potentials. Now, there are a few important parts of the retina. The retina coats the internal surface of the eye. There are a few important parts. These are the blind spot, oftentimes called the optic disc, the macula lutea, and the fovea centralis. All right, the blind spot, oftentimes called the optic disc, seen here, the macula lutea, seen there, and the fovea centralis, seen here. The idea is that the fovea centralis contains almost exclusively cone photoreceptors. When light comes into the eye, that light is focused by the cornea primarily. Well, it's the next day. We ran into some minor technical difficulties. Apparently, I've now learned that my camera can overheat and get so hot that it will cut itself off and say, you've got to rest for a period of time. So, I believe the last thing we were working on was talking about the inner layer of the eye. We had described the retina, which is the most internal layer, shown here in yellow, which houses all the rods and cones of the eye. Those are the photoreceptors. And then I believe we were talking about the optic disc, the fovea centralis, and the macula lutea. Okay. So the macula lutea, or the macula lutea, is like a donut of blood vessels that surrounds the fovea centralis. Let me say it again. The macula lutea surrounds the fovea centralis like a donut of blood vessels. And basically it's a super, like, vessel-rich area that provides the fuel needed, if you will, for the fovea centralis to function. The fovea centralis is jam-packed with cone photoreceptors, and cone photoreceptors, this is very important. Cone photoreceptors are those which give us our sharpest vision during daytime, so our vision is very unique to upper primates, and that extraordinary sharpness to our vision is provided by the fovea centralis. That's where the cone photoreceptors are mainly located. So the macula, the fovea are very important. The optic disc, on the other hand, is an area completely devoid of all photoreceptors. This is where the optic nerve connects to the eye. Through some weird variation on how photoreceptors are designed or set up in the retina is a fancy way to put it, but the way they're set up, because the optic nerve connects here, there can be no photoreceptors at that spot. So what you're going to do at home, I hope, is a blind spot test, and I'll be describing that to you in a little while. Alright, internally, you have the lens here. So we have the lens connected to the ciliary body. So the ciliary body holds the lens in place, and it can stretch or squeeze the lens depending upon what is necessary. And in front of the lens, there's what we call aqueous humor, a fluid that sort of balloons out the cornea. You can see how the cornea looks kind of ballooned out. And then behind the lens is a goo, like this gel called the vitreous humor. So aqueous humor in front of the lens, vitreous humor behind the lens. Perfect. And we'll be talking about tapetum lucidum here in just a little bit. Tapetum lucidum is a modification to animals' eyes that gives them an internal shining coat, like the internal coating of like a dog's eyes or what have you. Like you probably have seen when you drive down the road, the first thing you see when you see animals at night is their eyes shine. Tapetum lucidum is this shiny coating on the inside of an animal's eyes that gives them better night vision at the expense of sharp day vision. Whereas your eyes are quite dark in the back, so you have sharp day vision at the expense of better night vision. Alright, so let's talk about parts and pieces of the eye here. you will see this stuff again. You will see this again. Okay? So here is dissected images, and I'm going to be putting up a video for you to see a eyeball dissection. And then here is the models from lab, and you're going to be seeing those on your lab exam. All right, start here, and we'll just kind of work our way around, then we'll do the model. Actually, let's do the model, and then we'll work our way around. All right, on the model, what you see is the choroid here. So the choroid in brown, you can see all the blood vessels in it. So this is going to be the area that provides vascularization for the eye and kind of keeps the eye functional. You can see the retina. Okay, this is the internal, like this. Side comment. I don't care for this model. I really don't care for models overall, but I don't care for this model in particular. Let's just see here. So I believe that this would have been on top of that, and when that's on top of that, it looks like this, which makes this the iris and the pupil, and if that's the choroid, then when you open it up, that would be the retina inside of it, and this whole thing would be housed inside of the sclera, which isn't pictured. Okay. Alright, so the optic nerve's coming in here. Importantly, I'll point out the fovea centralis and the optic disc. This is worthy. A nice test question could be something like, identify this structure, fovea centralis. What would be surrounding it, macula lutea? What sort of photoreceptors would be prevalent here? Cone photoreceptors. Or alternatively, I could put the star way out here in the retina, and I could say, identify this structure, that's the retina. Part 2 could be what kind of photoreceptors would predominate here, and that would be rods. Okay, so the rods are kind of found in the periphery, whereas the cones are primarily found at the fovea centralis. Or alternatively, I could put the star on the optic disc. And I could say, identify the structure, optic disc. I could say, if there are photoreceptors, would there or would there not be photoreceptors here? And if so, what kind? Just to kind of play games with you. And the answer there would be, there are no photoreceptors on the optic disc. Okay, that's good enough. You can see the lens and the ciliary body there as well. Alright, let's start up here and work our way around. So what you have here is the shining internal coating called the tapetum lucidum. It's very beautiful in real life. It's a shame that you can't see it. Man, I wish we could be together so you could see this. It's very interesting. Regardless, you can see the lens and the vitreous humor here. So the lens kind of attaches to the ciliary body on the inside with little ligaments. But when you kind of shuck the oyster, so to speak, and kick that lens and vitreous humor out, it kind of pops out as a single unit like this. You get the lens and the vitreous humor. You can see a lens here. Here you can see the lens and the vitreous humor is in this cup inside the ciliary muscle, so you can see how it sits in there. So basically, when you cut the eye open, you take a probe and you stick it down inside of there and turn a circle, and then you can throw the lens out. Yeah, perfect. Alright, on the back of the eye, you can see the optic disc here, where it connects. Like you can see, if you look at it, like all of the retina, which is this brown stuff, kind of gathers up at the optic nerve. And make no mistake, the retina is loose from the rest of the eye. Like you can peel it off if you so chose to do so. It's like a layer of wet paper towel that lays against the choroid, basically, in this particular image. So you can see the optic disc here, where it would connect to the optic nerve. The choroid shown in black, sclera on the outside, ciliary muscle, the cornea would be on the outside of the eye, like this. The cornea would be attached here. Yeah. Yeah, I can live with it. Alright, very important. So, boy, I wish we could do lab. I would love for you to be able to see a microscope viewing of the retina. It's so fascinating, but I'm just going to throw this out here for you now as best I can. You're going to see this again. I feel this is very important, so you're definitely going to see this again. This is what the retina looks like in a photomicrograph. So if I were to come in to the retina, like here, and take a snip of retina and put it on a microscope slide, what you would have is, like, if this is the back of the eye, the pupil and the cornea and the lens would all be way out here. So light would come in through the cornea, go through the iris, be fine-tuned by the lens, come in, go through the vitreous humor, all the way back through the ganglia of the visual system. These ganglia are those which actually send messages to your occipital lobe. They have to go through these bipolar neurons. Remember we talked about bipolar neurons? They go through the bipolar neurons. The light then has to go through the nuclei, the rods and cones, until it actually hits the photoreceptors. All right, the rods and cones are going to be found here, right there, right there. And from here, the rods and cones are embedded in the choroid. All right, so the rods and cones are embedded in the choroid. Light goes from left to right in this image. It goes through this. So the rods and cones are embedded in the choroid. Here are the rods and cones. Here are bipolar neurons. Here are ganglia. I will put a pointer on this, and I'll say, what kind of cells are those? And you'll have to tell me. And I'm also going to point out to you that the choroid is darkly pigmented. Let me try again. The choroid is darkly pigmented with melanin to make it non-reflective in humans. Right? Our non-reflective choroid, it's got melanin in it. That's why it's so dark. In essence, prevents light from scattering inside of our eyes and increases our level of precision in terms of our way of seeing. Okay, so this pigmented choroid is very important. So choroid, which provides the blood supply, and it's pigmented to prevent light scatter. Rods and cones embedded in the choroid. They're nuclei. Bipolar neurons, then ganglia cells. Yeah, that'll work. All right. On to the ear. So in lecture, I'm going to go into more detail than you could ever want on this. For lab purposes, I'm mainly focused on the parts and pieces, although I will tell you a few other things. All right. What you have here is the pinna and the auditory canal terminating in the very important tympanic membrane, which you might know as your eardrum. Okay, the eardrum or the tympanic membrane is sitting there attached to the ossicles. These are called the malleus, incus, and stapes. Okay, malleus, incus, and stapes. Those connect to the oval window of the cochlea, and this is the cochlea. Not all this, but this is the cochlea. That pretty much is your entire hearing apparatus. Let me describe it to you in a little more detail. So the pinna collects sound waves and funnels them into your auditory canal. This is considered your outer ear. The outer ear is all of this. The outer ear terminates at the tympanic membrane, which is incredibly important in your eardrum. From the tympanic membrane to the oval window of the cochlea, this is your middle ear. The middle ear. The middle ear is tympanic membrane, malleus, incus, and stapes. The stapes is directly connected to the inner ear structures. This is very, very important for us, malleus, incus, and stapes. And the actual part of the ear apparatus, the inner ear, which is back here, that deals with hearing is this snail shell shaped part called the cochlea. Now there are more parts to the inner ear than the cochlea. These are called semicircular canals. This area is called the vestibule, but this is the cochlea. These two parts deal with equilibrium. The cochlea deals with hearing. Okay, let me say that again. The semicircular canals in the vestibule, they deal with equilibrium. They are not involved in hearing. Hearing is done exclusively by the cochlea. All right. Okay, now I'm going to point out one more thing before I move on, and I'm going to hammer this home in just a second. But inside of the cochlea, there's an organ called the organ of Corti, or the spiral organ of Corti, which is shown here. You're going to learn all about how this works in lecture. But for our purposes, I simply want you to know that you hear things with the cochlea, and the actual structure in the cochlea that allows you to hear is called the organ of Corti. So a nice question on the test would be, like, put a star on that. Say, hey, what is this? Cochlea. Part 2, what does it deal with? What special sense? Hearing. Part 3, what's the organ inside that deals with that? Organ of Corti. Yeah, that's great. Now, the rest of this is for equilibrium. So what we have here are these semicircular... Let me check something real quick. Yeah. What we have here are called these semicircular canals. I'd write that down if I were you. I appear not to have done that. I don't know why. And then back here, this is the vestibule. So we have semicircular canals here, vestibule here, cochlea here. And in purple, these are the organs that allow these structures to perceive motion. Now, or movement, you might say. They're all mechanoreceptors. All right, so let me describe to you how this works. Sound waves come in, they vibrate the stapes. The stapes sends vibrations through the cochlea. That means that the organ of Corti can perceive sound waves. When you move around, The vestibule and the semicircular canals allow you to perceive motion, but the way they do it is different. The vestibule, which contains an organ system called the maculae, basically allows you to perceive linear motions. Forwards, backwards, up, down, left, right. That's all done by the macula in the vestibule. So this is the vestibule containing macula. There are two of these, and they allow you to perceive linear motions. The semicircular canals up here perceive rotation. All right, rotation of the head is perceived by very special organs called the cristae ampullari, and they are inside of the semicircular canals. The cristae ampullaris in the semicircular canals, the macula in the vestibule, and the organa corti in the cochlea. That is important. Okay, that'll do for our purposes. We need to talk about conductive deafness versus sensory neural deafness. Now, I found a decent video. Well, I think one of my cohort made it. And I'm going to upload that for you, but I'm just going to describe to you sort of how this works now. Conductive deafness is a problem out here. Okay? If you've got conductive deafness, that means something over here is messed up. Everything inside of this is just fine, but it's out there. Like maybe your ossicles don't work right. Maybe your ear has impacted wax or something. Anything that keeps sound waves from being able to be conducted in, that is conductive deafness. Sensory neural deafness is here or there. Either the nerve is damaged or the organ of corti itself is damaged. And we can test this to find out how best to treat a person. Okay, the way we test this is by doing what's called a Retina test and a Weber test. These are normally done together, so you do one after the next. And what you do is you ring a tuning fork, ding, and you press it against your mastoid process. And what you're doing is you're conducting vibrations through the bones of the skull. Okay. Basically, you're vibrating the cochlea without the outer ear having to do any work at all. You're going around the outer and middle ear, going straight to the cochlea by vibrating the bones of the skull with a tuning fork. And if you can hear by doing that, then your neural apparatus is functional. If you can't hear without it, like if I ring the tuning fork and I hold it out here and I can't hear it, that basically means I have conductive deafness. I'm not getting sound waves through the outer and middle ear, ergo that's conductive deafness. If I can't hear anything and I ring the tuning fork and I pass those vibrations directly through the skull using that tuning fork and I still can't hear anything, that's a good way of identifying that you have neural deafness. Something with the organ of corti is not working. Okay? Something with the nerve is non-functional. And then we have this Weber test. So that was a Retina test. A Weber test, you ring the tuning fork, ding, and you put it on top of your head like so. Alright, and when you do so, what you're trying to do is you're passing vibrations directly through the skull again. And you're trying to see if one ear works better than the other. And the ear that works best, it tells you something in essence. Basically, I'm trying to figure out if one organ of corti works better than the other. And if they do work differently from one another, you might have some degree of problems. It's referred to as lateralization. I'm not terribly concerned at this stage with the Romberg test and the nystagmus. We're going to talk more about this in lecture. I will briefly discuss how smell works now. So what we have here are these cribriform foramina in these cribriform plates. So this is part of the ethmoid bone. We got cribriform plates, and I think I talked all about this when we did lab previously. These cribriform plates have little holes in them called the olfactory foramina, or cribriform foramina. And the essence of it is that you have these olfactory bulbs in the brain, and they lay down on top of these holes, the cribriform foramina, and they send bipolar neurons down into the nasal canal. So what happens is when you smell something, the chemicals from what you're smelling come up and hit these receptors on these bipolar neurons, and they fire off signals to the olfactory bulbs that go out to your temporal lobes, if I'm not mistaken, to allow you to perceive smell. All right, to allow you to perceive smell. This is very important. So it's all about these olfactory foramina and these bipolar neurons. All right, taste. Taste is fascinating. So what we have are on the tongue, you can see these little nodules, if you will. All these little nodules, these little spots. Those are not taste buds. Those are called papillae. There's lots of different types of papillae that we're not going to get into, but they're papillae all the same. And the idea with papillae is they are lined with taste buds, which are little bitty microscopic things. Like you can stop this video right now, go to the bathroom, stick your tongue out, look in the mirror, and you'll see the papillae. The papillae are lined with taste buds. Now the taste buds themselves look like this. They have a taste pore, gustatory cells, and then neurons back here in the back. And so what's going to happen is fluid saliva carrying chemicals comes in the taste pore, interacts with the gustatory cells, allows you to perceive flavors, and that sets off nervous transmissions that go to your brain as taste. All right, so taste buds are on papillae, and the papillae typically are on your tongue. And then basic taste sensations. So believe it or not, your tongue perceives different flavors in different locations. The idea is that you can perceive sour flavors on the sides of your tongue, sweet flavors at the very tip of the tongue, with salty flavors slightly behind that, and then over the back here you pick up bitter flavors. Indeed. So let's briefly discuss this. So sweet flavors tend to be driven by sugars. There are other things involved here as well, but typically driven by sugars. Sour things are acidic. So if it's sour, it's just simply a way of saying that it's acidic. Salty things tend to contain metal ions, like the chemical formula for salt is sodium chloride, so metal ions. Bitter things tend to contain medications like alkaloids, quinine, aspirins, all give bitter flavors. And then last but not least, umami, which is something we'll talk about a little more in lecture perhaps, but basically gives a sense of savoriness to food. It's a Japanese term. And that's it.